So we all know that cholesterol is part of the plasma membrane, but more importantly, the estroidal hormones or the sex hormones are made from cholesterol. So every day our body makes about 1 gram of cholesterol and we eat about 1/2 a gram. So our body is going to adjust their production of cholesterol depending on how much cholesterol we consume. So chylomicrons are basically triglycerides or fats, and they are composed of mostly fats or TAG, it's 92%, and just a small percentage of cholesterol. Chylomicrons are super small particles, and when digestion is occurring, the cholesterol that we ate is trapped inside the chylomicron, and these chylomicrons will travel to the liver where they're going to be processed. So basically, the chylomicron is the primary vehicle carrying cholesterol to be processed by the liver. So a cholesterol ring, which we either eat or our body makes, is never metabolized. It is going to be excreted into the bile acid first, which then goes to the intestine, and then it's going to be absorbed in the ileum, and then it's going to go back into the liver. So this whole entire process of the chylomicron traveling to the liver, and then the liver excreting the cholesterol rings into the bile acid, getting absorbed into the ileum, and then back to the liver. This whole cycle is called enterohepatic circulation. So to summarize, cholesterol from chylomicrons is going to be released in the bile acid. They're going to make their way to the ileum and then back to the liver. And this is essentially a recycling of cholesterol. So acetyl-CoA leads to the formation of cholesterol. And here's a picture that we don't really have to memorize, but this is just depicting cholesterol. So it's got 27 carbons and 4 cyclic rings. The tail end is going to make the hydroxy forms. The hydroxy groups are either going to bind to the 25th or the 27th carbon, and they can be called 25 hydroxy cholesterol or 27 hydroxy cholesterol. And these cholesterols are known as oxysterols. So the more cholesterol there is, the more oxysterols there are. But this isn't very too important. So remember that the lipid bilayer is very fluid. Cholesterol is lipophilic, so it can go through the membrane very easily. Some cholesterol remains in the lipid membrane and provides kind of rigidness in the membrane. So cholesterol is not spread evenly throughout the membrane. It's actually going to be scattered throughout. And in some places, there is a higher concentration of cholesterol, which makes the membrane less flexible and less permeable in those areas. To make one cholesterol molecule is going to take 18 acetyl-CoA, 36 ATP, and 16 NADPH. So acetyl-CoA comes from carbohydrates, lipids, and amino acids. The condensation of 2 acetyl-CoAs in the beginning right here is going to make them merge together. This acetyl-CoA molecules merge together, forming acetyl-acetyl-CoA right here. Then another acetyl-CoA molecule depicted right here is going to come in, and it's going to make a huge molecule called HMG-CoA. And this is the substrate of a very important enzyme called HMG-CoA reductase, as seen right here. So HMG-CoA reductase is going to convert its substrate into mevalonate, which is going to go into this really long, complicated process to make cholesterol. But we don't really need to know that. We just need to know that the HMG-CoA reductase is going to make the mevalonate, and then that is going to go and make cholesterol. So just note that HAMG-CoA reductase is a regulatory enzyme because it regulates the synthesis of cholesterol. Basically, if there's a high amount of this enzyme, there's going to be a high amount of cholesterol synthesis. So there's a transcription factor called SREBP, and this is short for sterol regulatory element binding protein. SREBP is a transcription factor located in the endoplasmic reticulum membrane, and it's going to bind to another protein called SCAP, and that is bound to another protein called INSIG. So basically, when there's high levels of cholesterols or sterols, we're going to have this whole protein inception thing, where basically all three proteins are bound together. So sterols are going to bind to this trimer around the SCAP area and stabilize this protein complex. However, when cholesterol is low, that means that the sterols are low, and therefore they won't be present to keep this entire trimer together. So what happens when low levels of cholesterol? So what happens in low levels of cholesterol is that SREBP bound to SCAP is going to move from the endoplasmic reticulum into the Golgi apparatus. In the Golgi apparatus, SREBP is going to be degraded, which is basically just proteolysis, and that degraded SREBP becomes active. The active form is then going to translocate into the nucleus, and there in the nucleus it's going to activate promoter genes and produce HMG-CoA reductase. And just remember that the HMG-CoA reductase right here is a regulatory enzyme, and it is going to regulate the synthesis of cholesterol. So basically what I'm trying to get at is when we eat a lot of cholesterol, there really isn't any need for this entire process right here, for the body to go through the process of degrading SREBP bound to the SCAP, and then using the active form to eventually produce cholesterol. Only when cholesterol consumption is low is when the body goes through that second process to make more cholesterol. So what happens when we eat something with high cholesterol? Well, our cholesterol is going to spike, and then the body is going to adjust by first shutting down the production of HMG-CoA reductase. So here is a glucose structure. It has 6 carbons and some hydroxy group. And then here we have galactose, which also has 6 carbons and some hydroxy groups. The only difference between the glucose and the galactose is that in the 4th carbon, right here and right here, for the 4th carbon, the hydroxy group for glucose is on the right side, while for galactose, it's going to be on the left side. So basically, they're enantiomers of each other. Also, just a little side note, but glucose's structure is in water is actually not linear like depicted right here. It's actually going to be a cyclic structure. So let's say you ate a starch. It is going to be absorbed once it's broken down to a monosaccharide or glucose. And then this glucose is going to go everywhere in the body. So let's say that glucose, a 6 carbon molecule, enters the red blood cells, while glucose is going to be phosphorylated into glucose 6-phosphate. And this is going to be, and when this happens, 1 ATP is going to be utilized. So then glucose 6-phosphate is going to get isomerized into fructose 6-phosphate, seen right here, another 6 carbon molecule. And this step right here, glucose 6-phosphate turning into fructose 6-phosphate, is actually going to be reversible. So fructose 6-phosphate is then going to get phosphorylated using 1 ATP molecule to make fructose 6,1,6-bisphosphate, and this is a 6 carbon molecule again. So just a side note, if both of the phosphates on fructose were on the same carbon, it would be called diphosphate instead of bisphosphate. So basically the fructose 1,6-bisphosphate just means that there's a phosphate on the first carbon and the sixth carbon. If both the phosphates were on the same carbon, it would just be fructose diphosphate. Anyways, so aldolase right here is going to split fructose 1,6-bisphosphate, a 6-carbon molecule, into two different molecules, DHAP, which is a 3-carbon molecule, and then G3P, which is a 3-carbon molecule. Okay, so these two molecules, G3P and then DHAP, can be isomerized into each other reversibly. So 3GP can turn into DHAP and DHAP can turn into G3P, but we mostly see DHAP getting isomerized into G3P more often. So in the next half, we're going to see G3P right here going through this whole process to become pyruvate, producing 2-ATP. But remember that this is just one half of the process. The other half of the process is when DHAP isomerizes into G3P and repeats this entire process, making another 2 ATP. So in total, we're going to have 4 ATP molecules made and 2 ATP molecules used from all these steps over here. So basically, we're going to have 2 net ATP made. Also note that we're going to get 1 NADH from this half of the cycle and another one from DHAP cycle. So altogether, we're going to get 2 NADH molecules made. So when there's enough energy in the body, ATP is going to bind to phosphofructokinase, and it's going to inhibit phosphofructokinase, and this is going to serve as a negative allosteric regulator. On the flip side, when ATP is being used rapidly by the body, ATP gets dephosphorylated into ADP, and then ADP is going to get converted into AMP. AMP is going to bind to phosphofructokinase and serve as a positive allosteric regulator. So going back, red blood cells do not have mitochondria, so they can't make ATP. Instead, pyruvate right here, made in the red blood cells, is then going to be converted into lactate through the use of one NADH molecule, and that lactate is going to travel to the liver, which then turns the lactate into glucose, and this is called gluconeogenesis. So when there is oxygen available, pyruvate is converted into acetyl-CoA. And just a side note, but pyruvate is 2 carbons, while acetyl-CoA is 3 carbons. Pyruvate is 3 carbons, while acetyl-CoA is 2 carbons. That extra carbon loss just becomes carbon dioxide, and then this whole process, 1 NADH is formed. And then for fermentation, pyruvate is going to be converted into acetaldehyde and then into ethanol using 1 NADH molecule. So if you've ever gotten your blood drawn, you'll notice that when you get these tubes, you're going to get these tubes coated with EDTA, and this prevents your blood from coagulating. And then they're also going to put an enolase inhibitor, kind of like fluoride, into that tube as well. If enolase is not inhibited, then glycolysis will continue. But as long as enolase is inhibited, then glucose 6-phosphate will accumulate, inhibiting hexokinase itself. And this, right here, then this is going to, glucose 6-phosphate is going to accumulate, and this serves as a inhibitor of hexokinase. And when hexokinase is inhibited, then this causes glucose to remain unmetabolized in the blood, and this is what we want when drawing blood. So here is arsenic right here, and here is phosphate. As you can see, arsenic and phosphate look very similar to the point where G3P dehydrogenase cannot distinguish between the two. G3P dehydrogenase will bind to equal affinity to both phosphate and arsenic, and when it binds to arsenic though, it will make 1-arseno-3-phosphoglycerate instead of 1,3-bis phosphoglycerate, which leads to a whole cascade of events where there's going to be a net zero production of ATP in the red blood cells, which is eventually going to lead to death. Alright, that was it for this lecture. I'll see you guys next time.